specific requirements for setting up an animal cell culture. So here there are some prerequisites, so which are very important for setting up an animal cell culture laboratory. First thing is they should be free from the pathogens and microorganisms. So completely we have to check at every time interval, like within like every 1 week or 10 days we have to fumigate. Okay, fumigation is nothing but we have to release the fume so that even in the on the walls the fungus everything should get killed. Then all these instruments also we have to clean regularly before use and after use also, so that we can prevent the growth of microorganisms. So here we have to grow the animal cells in the controlled condition. So controlled condition means we should provide all the requirements for the animal cells that are there in the body so that they can grow well. So what all we have to provide? First is the medium. We will have special dishes. Okay, special no, like petri dish and for microorganisms. So in the same way, we will have some culture dishes for the animal cell culture. So which will be mostly sterilized. Okay, so we should have that culture vessels or dish. Then the conditions should be aseptic. Then the media we have to provide which is suitable for the growth of specific animal cell line. So that medium should consist of amino acids, carbohydrates, vitamin, minerals, growth hormones, then growth factors, and the gases. Okay, like carbon dioxide and oxygen. All these are important, which are suitable for the growth of specific cell line. And also we have to maintain pH using buffers, then osmotic pressure, temperature, everything should be maintained. So here the cell lines will be of two types, mostly adherent cell lines or monolayer cell lines, and some are suspension cell lines. So adherent cell lines are nothing but which will attach to each other. Like if you see our skin cells, they are like a monolayer, right? All the cells are attached to each other side by side and they are like a single layer. So they are called monolayer culture. So normal healthy cells like skin cells and other tissue cells, they will be in monolayer. Then suspension cells means the cells which will remain in the suspension. They won't bind to each other. Example, blood cells. So blood cells will be there in the blood. They won't attach to each other, right? And pathogenic like cancer cells. So the normal cells when get transformed into cancer cells, they lose this capability of adherence. Therefore, they become suspended. Okay. So for suspension cells, we have different type of flasks. For monolayer cells, we have different type of flasks. And the medium also will differ according to their requirement. And what are the instruments? First one is laminar flow hood. So laminar airflow also called as, so it is the area where we have to actually perform all our experimentation. So this will provide aseptic condition. So here we have three classes, class 1, class 2 and class 3. So this is how it looks like. You might have seen in the microbiology laboratory and all. So usually in the microbiology we use this class 1, and class 2 is mainly used for the primate cultures, radioisotopes, cell cultures, all these things. And class 3, it is mainly used for the human pathogens. Okay, it is the use. So, but basically class 2 is mainly for the animal cell culture. Then the laminar airflow protects the working environment from the airborne contaminants. So here you will have a filter which is called HEPA filter through which the air will flow. Therefore, the air will be filtered and it is sanitized. Okay. So there will be no microorganisms present in the air also. Then we have to clean that bench with ethanol. Okay. So before starting the work and after starting the work also, we have to always make clean and sterile environment. So we have to clean that benches with the ethanol. Then, so it will have cell culture hood layout. So you can see here, yeah, as I told, we have to use 70% ethanol. So everything like, so air will be definitely filtered. Then only the workbench should be cleaned. Then all the arrangements of items within the cell culture hood should adhere the right-handed convention. Okay. So here the pipette also should be cleaned with the tissue paper with 70% ethanol, and all the reagents and media should be filtered or you call what is that, autoclaved. Okay, some like the glass vessels, all these things, equipment should be autoclaved, whereas the plasticware and other chemicals, okay, chemicals should not be autoclaved. Media can be autoclaved, but proteins like serum or some enzymes, all this should be filtered. Okay, so all this should be done before starting the cell culture. Then there should be a rack where we have to keep our tubes. And for the waste, once you have to all the tips and all for the pipetting, so they should be discarded. So that should be, we should not remove our hand outside. So even our hand should be cleaned with the ethanol before placing our hand inside the laminar hood. So everything, once you place your hand inside, you have to do all the experimentation, then only you should take your hand out. Again, once you come out, you have to again wash your hands with the soap, and again you have to go back, then you have to wash it with ethanol, and then only you have to start the experimentation. Okay, so everything should be inside, nothing should be outside. Okay, even the waste jar, a small jar kind of thing also, we have to keep it inside on. Then incubator also you have to place it accordingly. It should be nearby to the laminar airflow. So incubator is specially designed for the cell culture where the moisture and the temperature, and it will have a carbon dioxide cylinder. So 5% carbon dioxide should be supplied continuously. So all these should be present. So all these should be maintained. You have to check whether water is there for the humidity purpose, and the gas is present in the cylinder or not. Okay, the temperature is fluctuating, or whether it is, because especially in summers the temperature will rise. Okay, so everything, the complete room should be air conditioned mostly. Okay, so but in some cases, like, we have to check everything, like not only the cell culture, but all these factors will impact the growth of the cell. So if it is the temperature is fluctuating, we have to check. If the gas is fluctuating, we have to check the pH. Okay, so here the incubator is called as carbon dioxide incubator. So because we are supplying the carbon dioxide, 5% carbon dioxide using a cylinder. Okay, then for the storage purpose, so where we have to store liquids like media, reagent, drugs, antibiotics, then means some should be stored in the fridge, some should be stored in the room temperature. Okay, media bottles, glass pipettes, glassware, plasticware, everything, we should have a proper storage place. Everything should be stored in the aluminium foil wrapped. Okay, some should be, if they are light sensitive, you have to place it in the dark place. If they can tolerate light, you can keep it in the light. So all these you have to check. Then refrigerator. So 2 to 8 degrees centigrade. This is the temperature where we can store the reagents, all these enzymes and all this. So sometimes you will have cold rooms or minus 80 degrees. So where you will store the cells mostly, okay, for long term storage. But every day if you are working, so you can store in the normal refrigerator. So that is next is freezer. Okay, freezer is -5 to -20. Then you'll have deep freezer -80. So this is according to your requirement. Suppose, see here, suppose I want to do an animal cell culture experimentation. Today, if I want to start, okay, I should not stop the experimentation till some 1 month. Okay, I have to perform all the experimentation within the 1 month. Okay, suppose because after 1 month I am going to some vacation or something, I cannot work. Then what I can do? I can store these cells, I can culture them, I can store them in -80 or -20 for 1 month. -20 you can keep for 1 month like that. But I want to store it for more, like 6 months, like that. So you can store it in the -80. Okay. So here, once I have to start, so you have to pre-plan the complete experimentation. Culturing, how many days it will take. Once I culture, how to plan the experimentation. Everything you have to plan, and then only you have to revive the cells and you have to start. Because especially in India, Indian scenario, we'll have like rain. If it is a rainy season, there is a lot of contamination with the fungus. If it is summer, lot of temperature fluctuations, all these things. So we have to maintain everything properly. If any one of the factors are not maintained, so the complete cells will die, and it is again difficult to procure the cells. Okay, so that's why everything should be planned accordingly. Then cryogenic storage is nothing but, so here um, cell lines, they usually, if you culture for many generations, that is called as passage number, their genetic, the genes will get mutated, genetic instability will be removed. Okay, then if you want to store it for longer period times, okay, there will be cryogenic storage. Okay, so here there are two main types: liquid nitrogen storage and vapor phase and liquid phase. And vapor phase system minimizes the risk of explosion of this cryo storage tubes. So you will have a separate cryo storage tubes where you have to add the cells. Then you have to add DMSO, that is a detergent. Sorry, not detergent, that's a chemical, dimethyl sulfoxide. So the DMSO and methanol. So that will form a layer. So which will protect the cells from the freezing, and it will maintain them live for longer periods of time. And this should be placed in the liquid nitrogen. Okay. Then water bath. So water bath is to incubate the samples. Okay. So sometimes during the what you call protocol, you have to again incubate the cells or incubate the samples. So then you have to use the water bath. Okay. Then centrifuge. So once you take the cells, it will be in liquid form. Now you want the cells to revive. So revive is nothing but, suppose there will be flask, okay, animal cell culture flask. So in the flask, it has some capacity where you can grow only 1 lakh cells in that flask. But these cancer cells, like 1 or 2 days only, it will, after completing 1 lakh, again it will be continuously growing 2 lakhs, 3 lakhs, like that. If you keep like that only, so the media will be completed, okay, depleted, okay, and the waste will get accumulated because the metabolism will be taking place in that. So in such conditions, the pH will alter and the cell starts dying. So therefore, during that process, we have to split the cells into another flask. So during that time, you have to use the centrifuge. Is it clear, this point? Or any doubt till here?
No, ma'am.
Okay. So splitting is nothing but, suppose in my flask I have 1 lakh cells. Now today I have 1 lakh, so it is completely filled. That means we call it as 100% confluence. That is 100% the cells are filled in the flask. If I keep it like that only, by tomorrow they will become 2 lakhs. And what happens? The medium which I added in the flask, that is that is only for 1 lakh cells. Now when they become 2 lakhs, what happens? Half of the cells starts dying, and already the waste will be produced by these 1 lakh cells that gets accumulated, that produce toxins. So all this happens. So that is waste of money only, right? I am culturing the cells for some purpose, some research. So therefore, what I will do today only, I have to split this 1 lakh cells. So what I will do, first I will add trypsin, which is the enzyme, to this flask. So what this trypsin will do, it will remove all these cells which are attached to the bottom of the flask. Once, just I have to keep it only for 1 minute. So it will remove all the cells. All the cells will come into the media. Now immediately I have to add PBS, phosphate buffer saline. So which will neutralize this trypsin. Okay. Now I will take this media. I will centrifuge this media so that all the cells present in the media will come to the bottom. And I will remove the top portion, supernatant. To this pellet, I will add the fresh media. Again, I will add 1 ml of fresh media. I will mix it thoroughly. Now what I have? I have 1 ml of fresh media with 1 lakh cells. Got it? Now what I will do? I will add this 1 ml into 2 flasks instead of 1 flask. Or I can sometimes add in 4 flasks also. It's my wish. Okay. So suppose I am adding it because suppose sometimes what happens, second Saturday, Sunday, like that holidays will come. Means lab may not be open. So what I can do, I can add them in the 4 flasks. Okay. Now 1 ml, I'm adding in like 250 ml, 250 ml, 250 ml, 250 ml in 4 flasks. And I'm adding fresh media. No, without any cells, only fresh media. Like 4 ml in each flask. Now, how many cells are there in each flask? Can anyone tell? I have 1 ml of media with 1 lakh cells. Yes. Initially, I have 1 ml of media with 1 lakh cells. Now, I'm adding, that is, I'm distributing that into 4 flasks, each with 250 ml. So, how many cells are there in each flask now? Yes. 25,000, right? 25,000 into 4. I have 1 lakh cells, right? So I am distributing this 1 lakh cells into 25,000 cells each in each flask. That is 4 flasks. Now by tomorrow, this 25,000 will become 50,000. Then 50 will become 1 lakh. So I have 2 days time to again achieve 100% confluency. So within this 2 days time, I can plan some other things, right? Is it clear?
Ma'am, once again, can you tell?
So did you remember like when I centrifuged? So initially I got 1 lakh cells. 1 lakh cells, from 1 flask I took 1 lakh cells. To the 1 lakh cells, I am adding fresh media. Okay, so I have 1 ml of media with 1 lakh cells. Is it clear till here?
Yes, ma'am.
Okay. So suppose I add this 1 lakh in another flask. What happens? Again, it will become 100% confluent. I don't want that, right? I want some time so that these cells again become 100% confluent. So what I am doing, this 1 ml, instead of adding in one tube, one flask, I am adding in 4 flasks. So once 1 ml, I am equally distributing in the 4 flasks. 250 ml, 250 ml, 250 ml, 250 ml. 1 ml means 1000 ml, right? 1000 microliter, sorry. 1000 microliter. So I am adding 250 microliter, 250 microliter, 250 microliter, and 250 microliter. Okay? Is it clear?
Okay. Yes, ma'am.
In 4 flasks. So what is happening? These 1 lakh cells are distributed equally. 25,000 cells in one flask, another 25,000 in another. Like that, 4 flasks with 25,000 cells in each. So today I added 25,000. So by tomorrow, this 25,000 cells in each flask will become 50,000 cells. And after one day, day after tomorrow, this 50,000 will become 1 lakh. So now I am getting 2 days time, right? So once I add media and cells, we can change the media like after 2 days also. Okay. So I'm getting 2 days time to get 100% confidence. Again, 1 lakh cells means it completely the flask gets filled. Is it clear till here?
Yes, ma'am.
So for this purpose, we use centrifuge. Like for every step, we use centrifuge here. Then cell counter is nothing but now whatever I am telling, like 1 lakh cells or 25,000 cells, how will I know? Yes, I cannot see them with, I can see under microscope. Cells are there or not, whether the cells have died or not. But I can't count 1 lakh cells or 2 lakh cells. Some big flasks, they will have some n lakh cells. Okay. So now you will have cell counters. So where we have to use 1 microliter of sample and we will have a counting formula so that we will add this small sample, like 1 microliter onto that, and then we will count and we will calculate according to the size of the flask, and we will get the how many number of cells, whether it is 4 lakhs or 1 lakh or 2 lakhs, like that we will get. Then
So,
in
ma'am,
what, sorry,
it is accurate.
yeah, it will be mostly, see, one or two cells may alter because it is manually done. Some will be also like, some will be also done automatic also. If it is manually done, few number of cells, but mostly it will be accurate only. Means
Okay.
it comes with the experience. Okay. So like
Okay.
accuracy. Okay. Then inverted microscopes. So inverted microscopes are nothing but, so we know normal microscopes, whether you have figure or, yeah, here it is. Sorry. So here you have. So can you see? It is somewhat different with the, when compared to normal microscope. Is it right?
Yes, ma'am.
So what is the difference here? Can you guess? Like you might have used micro in the microbiology microscopes.
Yes, ma'am.
So where you will have the stand to place a slide, that will be at the bottom and the eyepiece will be at the top, right?
Yes.
But here eyepiece is at the bottom, but the stand is at the top.
Yes, yes.
So that is inverted microscope. So why we have to use this for the animal cell culture is the flask, whatever the flask will be not vertical. It will be horizontal. Okay. So is there any image here? The flasks. Yeah. Here you can see cell culture flasks. Can you see here? They'll be in the sleeping position. Okay. So that brown color one, shaded one, are the cells which form a layer at the bottom edge. Is it clear?
Yes, ma'am. So, directly we have to keep that flask on the microscope.
Yeah. So exactly in this position only we have to keep on the microscope. Then what happens? That bottom portion can be seen by this eyepiece.
Okay.
Is it clear? If you have eyepiece at the top, that has to go through, the image has to go through one more glass layer, right? Because the cells are at the bottom end. So now, because the cells are at the bottom end, you have eyepiece at the bottom side so that you can directly see the cells without any barrier. Got any idea?
Yes, ma'am. So because of the flask only, they have invented this type of microscope.
Yes,
Okay.
because the cells here are monolayer cells. Any animal cell you take, you cannot grow, like some are suspended, like blood cells and cancer cells, but they are rare, right? So when compared to all other cells, like all other tissue cells and skin cells and every cells, so they will form usually monolayer. And always this monolayer will be formed at the bottom of the flask, but not at the top. Okay, so that's why they have developed this kind, like Lawrence Smith. Actually, he invented this inverted microscope. For the suspension, it will be different, because suspension means they'll be in the flasks are different, and all the other protocols also will differ. Okay, and
Yes,
even
ma'am.
cancer cells also. Sorry, yeah, yeah. Ask any doubts?
No, ma'am, no.
No, okay. So even all cancer cells are not suspended. Only some transformed cells will be suspended. Most of the cancer cells also we use this monolayers only. Okay, and how will you know whether the cells are live or dead? So without doing any experimentation, simply by looking at the flask, also you can tell. So every day, first, when you enter the cell culture lab, first you have to take the flask. You have to see under microscope. That is the first step, because you will check whether the cells are dead or alive. So can you guess by how? Can we guess any idea?
No, ma'am.
Okay. So because the live cells, I told you monolayer cells. So whenever they are dead, it may be because of any reason, they will float. Okay, float in the medium. So
Okay.
usually one or two floating cells will be there because our handling or pipetting, anything. So nobody is perfect, right? How many experiments you might have done, but still during pipetting some cells will die. But if all the cells are floating, or 70% of the cells are floating, so there you can expect some problem, maybe contamination, or you have to change the media, or confluency, or pH change, or temperature fluctuation, or moisture change. So like that, you have to rule out each and every thing. Okay. So then you have sterilizer. So sterilizer is nothing but where you will sterilize the material. Okay, as like autoclaving. Okay, you will also have other sterilizers like under pressure. So the steam comes. Usually we use autoclave in the laboratories. So these are the equipment we usually have in the animal cell culture. In high-end labs, you may have high-end microscopes and all these things, but in normal lab, basic instruments are these. So why we need to do the animal cell culture? So what are the applications? So one is for the manufacture of viral vaccines and other products of biotechnology. This is again a large scale production. So whatever just now I explained you, that is in a normal laboratory setup. But most of them, so only these processes only, they'll get automated in the large scale. Then culture of human stem cells can be used to expand the number of cells and differentiate the cell. So for organ transplantation, for the artificial organ production, all these things. Then also stem cell culture is used to harvest the molecules and exosomes for therapeutic purpose. So exosomes are the normal circulatory vesicles in the blood. So they will have some receptors and all. Nowadays, exosomes are mostly used in the therapeutic development. Then we have recombinant DNA technology. So rDNA technology, where we can actually synthesize enzymes, synthetic hormones, immunobiologics like monoclonal antibodies, lymphokines, interleukins, all these things, anti-cancer drugs. Then also we can use bacterial cultures for the production of so another products like proteins and all. So for that we use this animal cell cultures. Then erythropoietin production. Okay, this is another hormone. Then somatic embryos. So yesterday I told you Dolly and all. Then for agriculture purpose, for the production of milk. So once you culture in the laboratory, you have to, this is called in vitro. Then this should go to the next level, which is in vivo. In vivo means in the animals itself. First step is you're doing in the lab. If it gets success or if you find it as promising results, then you will go to the next step. Okay. And mostly the advantage is here, we are not using or killing any animal. So most of the experiment you have to do here in the animal cell culture. Then only we should go for the animal experimentation, because even they have some ethical concerns. We cannot kill as number of animals as we want, or we should not perform like that. There are some specifications, there are some limitations. So most of the experimentation should be done in the in vitro level itself. Okay, so this is an best tool for the teaching of cell biology. So we are, while we are doing animal cell culture, we can understand more amount of details about the cells. So this is about animal cell culture. Then we will discuss a bit about transgenesis. Okay, so transgenesis is nothing but breeding. So it's nothing but a cross breeding of animals. So previously, even like we used to do cross breeding, we used to get hybrid varieties, all these. But after the emergence of biotechnology, there are some protocols how we have to perform and what is the purpose of doing that. So during 1970s, the first chimeric mice, chimeric means hybrid, was produced. The cells of two different embryos of different strains were fused to form an embryo, and this embryo was developed into a chimeric adult. Okay, so why we are doing that? So to produce something like transgenic animal. So the animal is called as transgenic animal. So it is for increase of the production of something, like if it is sheep for the production of wool, if it is cow for the production of milk, if it is any other thing for production of some meat or something else. Okay, like we have already transgenic animals which are successful are rats, rabbits, sheep, pigs, birds, fish. Okay, and two other main techniques were also developed, which are called retrovirus mediated transgenesis. That means virus is used to mediate the transfer of genes, and embryonic stem cell mediated gene transfer. So we can use the embryonic stem cells for the gene transfer. So now we will see this transgenesis. It is the process of introducing a gene, which is called transgene, from one organism to the genome of the another organism. So what is the purpose? To express a gene or a new property. So the first transgenic organism was created in 1974 by Annie Chang and Stanley Cohen, where Staphylococcus aureus genes were expressed in E. coli. Then 1978, yeast cells, which are eukaryotic, they undergone gene transfer. Then in 79, mouse embryos were transferred. So like that, slowly, one by one, transgenic organisms were developed. Okay. So here, micro injection of DNA directly into the cell was done. So directly they will have a syringe kind of thing. They will directly inject the genes into the embryo. So that was done. So then electroinfusion. So electroinfusion means we will give small electric shocks so that small pores will be formed, and we can introduce the DNA so that the DNA can pass through the cell wall and into the cell. Then we will use some ice, cold conditions. Then it will close the pores. Okay. Likewise, many techniques were developed. Then gun shooting means a shotgun method. So like that, many techniques were developed. So the first transgenic animals were intended for genetic research, but later they are used for the expression of some proteins. So here, what are the methods? First is, so there are three important methods. First is pronucleus microinjection method. So here, I will explain you the figure so that it will be easy. So here, this is the transgene. So the gene which we have to introduce. And this is male mouse, and so the shaded one is female one, white color one is male. So they have fertilized the gametes and will get
Ma'am,
one cell. Yeah,
use the marker, ma'am.
marker. Okay. So here, can you see like this? This one is female, green color one, which I highlighted. Okay, this red color, this is male. So after fertilization, one cell embryos were developed. So then, meanwhile, they have taken the gene of interest, okay, which is called transgene. Now they introduce this transgene into the embryo. Okay, they have removed the embryo nucleus from the embryo, and this gene was introduced. Now, after that, so this was introduced into the surrogate parent. Okay. So here we have, sorry, just a second. Okay. So here, once the embryos were developed, okay, one cell embryos. So here this transgene was injected into the male pronucleus. Okay. And okay. Now this embryos, injected embryos, male pronucleus, were injected into the pseudo pregnant female. So we are artificially making it a pregnant. Okay. So from this male pronucleus, which is having the transgene, so that embryo is there, no? So that is introduced into the surrogate mother. Okay. So from surrogate mother, it will give, it is sterile. Okay. Normally it is sterile, but we are injecting it from the outside. So it is giving rise to a male mouse, which is having the transgene. Now this will become the transgenic founder animal. That is, it is now the first animal with the transgene. From this animal, you can actually produce transgenic animals. Is it clear?
No, ma'am, I have not understood.
Just a second. So, first, have you understood this? Or else from the text itself, I will just check it. So it was first described by Gordon and colleagues. Here, male and female pronuclei, they have taken. So male pronucleus and female pronucleus. Okay. Now fertilization took place. Okay. The transgene may be injected into this either of the pronuclei. So here they have introduced in the male pronucleus, or you can also introduce in the female pronucleus. Okay. Then here the transgenic integration into the genome of the founder animal. So here what is happening? Sorry, just a second. Yeah. So here, can you understand the symbols of male and female? A round with a arrow at the top is female, and a round with plus symbol is a male. Okay. So here what is happening? We have pronucleus of female and pronucleus of male. So both are fertile. Fertile means which can give birth. So now after fertilization, one-celled embryos were collected. Now you have embryo. Okay. And in the embryo, so initially this pronucleus. So what is either male pronucleus or female pronucleus? We can introduce this transgene. Transgene is nothing but the foreign gene. So that gene can be introduced either in the male or in the female. So here what they did, the transgene was injected into the male pronucleus. Okay. So once you got the embryo, that male pronucleus with transgene and female pronucleus. So they got fertilized and you got an embryo which is having the transgene. Now this embryo was introduced in a pseudo pregnant female. So here actually the mouse was not pregnant. So by introducing the embryo, it is like a surrogate mother. We are making it pregnant. And that mouse is by itself, it is sterile in nature. Now it will give rise birth to a mouse which is having the transgene. So either of the mother or father are they are actually not having any transgene. This is all experimentation. Then now the mouse which is born, this is having the transgene by birth. So it's called transgenic animal. Now this is the founder animal, what we call. From this mouse, actually the gene will get transmitted to the next generations. All transgenic animals will be produced. Is it clear?
Yes, ma'am.
Okay. Then another method is embryonic stem cell mediated gene transfer, where we have embryonic stem cells. So we are adding DNA to the embryonic stem cells. So whether any DNA like insulin gene, suppose we are introducing the insulin gene into the embryonic stem cells, and all the embryonic stem cells won't take up the gene, only some of them will take. So we have to select. Yesterday I told you, no, we have to select the cells. So we have to select the cells with the gene of interest. Then these cells should be introduced into the blastocyst. So what is a blastocyst? After embryo is formed, it will have single cell. So that will become 2, 2 will become 4, 4 will become 8, like that it will increase in number. It will form a inner cell mass of some number of cells. That is called blastocyst stage. So which will be actively dividing. So we have to introduce this embryonic stem cells into the blastocyst. And this embryo, we have to place it back into the foster mother. Foster mother is nothing but a surrogate mother. Okay. Now this surrogate mother will give rise to another mouse which will be transgenic. We have to check whether the transgenic mice is having this gene or not, for the presence of gene. If it is there, if the gene is present, we have to always mate this transgenic mice with another transgenic mice to get the true population. Is it clear?
Yes, ma'am.
Okay. Another method is this pronuclei in the fertilized egg. So that pronuclei should be injected with the gene of interest using any vector, like we did for the mouse in the previous step. Then this pronucleus that is the embryo, single cell embryo, that should be introduced into the foster mother, and then it will give birth to a transgenic mice. So any transgenic animal also, and we have to do the next steps are same. Is it clear? So these are the two steps. Then we have retrovirus mediated steps, where this is the donor female. So donor female, it is already having, it is a pregnant female. So it is already having embryo. So we have to remove that embryo, which is 8 cells stage. Now what we are doing, we are infecting the embryo with a retrovirus. Now what this retrovirus is having, retrovirus is already having the transgene. This gene is already present in the retrovirus. Now this retrovirus will infect this embryo and it will introduce this transgene into the embryo. Okay. Now this embryo, we have to keep it back in the female. Okay. Now it will give birth to, it can be one, usually in animals, we can expect 3 or 4 offsprings. Based on different animals will have different number of offsprings. So among the population, we have to check which offspring is having this transgene. So that will become the transgenic animal. So again, we have to like perform mating of this transgenic animal with another transgenic animal, so that we can get the offsprings having the transgene. So they are called as transgenic animals. Is it clear?
Yes, ma'am. So for every animal, we are having these three. We can follow these three types of principles, ma'am. For
Yeah.
transgenic animals.
Yes, the method is same, but we have to choose which method is suitable for which animal.
Okay.
All these methods were already, they are proven. Like for small animals, the methods will be different. For larger animals, the methods will be different. And even these are some initially developed methods. Now even advanced methods also might have come. So which will reduce the time and all. Okay.
Okay.
So these are the
Okay.
different methods. Okay. Then nuclear transfer. So in the nuclear transfer, we are taking the tissue donor, which the cells from the animal to be cloned are maintained in the lab, so that they do not grow or divide. We should not make them divide. And from that, the nucleus should be removed. That is somatic cells. Now, we have to take another sheep, like for the dolly, how we have done. So, we have to remove the egg cell from another donor and nucleus should be removed. And this nucleus, somatic nucleus, should be introduced into the egg cell of the donor cell. Now, this again should, this embryo which is dividing, that should be kept back in the surrogate mother, and this cloned animal will be born. So this is example is dolly, which we discussed yesterday. Okay. So there are many examples, but you can choose any one of the example and you can explain. Okay. Okay, let us stop here. So till nuclear transfer, I have explained. We will continue with the other topics tomorrow. So we have transfection of gametes, artificial chromosome method, and testis cell transplantation method, and other recent methods also there, like lentiviral transfer of oocytes and zygotes and chimera generation method. And we will discuss one or two transgenic animals because there are many examples, but I will discuss only two like that so that time permits. Okay. Any doubts?
Okay, ma'am. No,
Okay. Okay. Thank you.
thank you, ma'am.